Okay, so thanks, Beck. So, hi everyone. Some of you may have heard a bit of news last night in the Herald Sun, and it's beginning to hit the media now. So this is incredibly exciting. So I'm going to talk today about a new drug screening platform for MND. And this is a Cure for MND Foundation initiative, and I'd just like to point out this is a world first combination of technologies right here in Melbourne. So I think let's start with what is the problem with MND at the moment? Why is the medicine cabinet not full? Why are there limited options? And I think of MND as a very challenging disease, and it's largely due to three core problems. So for those of you familiar with the disease, this disease is never the same in two individuals. This disease has a very different clinical pattern, and this is quite a useful diagram from a recent review paper from Paul Torman and colleagues, in which they've shown that MND has bulbar onset, it has arm onset, limb onset. And also there are other forms of MND, such as the flail limb and a slowly progressive form called PLS. So this is the first challenge, that MND is clinically heterogeneous and variable. So how do we model that in the laboratory? The second challenge we have as scientists is the causes of MND become increasingly more complex, and this is a useful graph here showing that in the case of inherited MND, which we know about 10% of cases, the number of genes has almost increased exponentially in the last couple of years. And this is a graph showing that the rise in the number of these different gene defects which can cause MND, and even now there's another cluster of genes at this end. So MND is becoming increasingly genetically complicated. Now, the genes point to disease pathways within cells that underlie their susceptibility to disease. So the second problem we have, or third problem, is that MND has increasingly complicated biology. And this is a useful diagram for Matthew Kinnon's review paper here. And what we can see is this is a motor neuron, and we can see there are several events, disease events in this cell, which are contributing to the disease. Perhaps they're all contributing. But not only that, we have cells, surrounding cells such as support cells, astrocytes and microglia, which are also contributing to the disease. So what is the solution to all of this? And I think that by establishing what we call a high throughput drug screening platform for MND, we can begin to circumvent some of these problems. So what is this platform? This is a world first drug screening platform dedicated to MND. So this is unprecedented. I wish to point out that this is a $5 million project which is being generously co-funded by the Victorian State Government, as announced, Cure for MND Foundation, and also the Balkan Company. The objective of this drug screening platform is simple, and that is it aims to rapidly discover and screen therapeutic agents using MND patient-derived cells, so this is the key, to identify priority agents that can be then advanced to potential clinical trials in Australia. So how does this pipeline work? This is an overview. So the starting point of this is always the patients. So in collaboration with neurologists, we can have access to patients, and we can use a technology which is called stem cell technology, IPSC. So I'll talk about that in a moment. So what this involves is taking a skin sample, creating some skin cells, and we're able to use some clever tricks to turn these cells into motor neurons, which you can see here, and we can put them into these high-density assay plates to do experiments. So once we have these cells, we're able then to move into what we call HTS. So HTS is high throughput screening. This is a drug screening platform. This is a new system which has just been launched, and we're able to introduce commercial drug libraries. Ultimately, we will generate several positive leads and hits from this screen, from thousands of drugs, and then these hits can be advanced to preclinical trials in animals, which you'll hear a lot about today from the grant recipients. But more importantly, we bring this back to the patient, so we bring this forward to clinical trial. So let me just talk a little bit more about the various stages of this platform. So if we start at stage one. Stage one involves a Nobel Prize winning technology known as IPSC, and this stands for Induced Pluripotent Stem Cells. So this just means we're able to make stem cells from patients with MND. And this is just an overview of the process. So as I mentioned, patients were able to obtain a skin biopsy, create skin cells in the dish, turn them back into stem cells, and then drive them into motor neurons. And this is actually real-life examples from our lab at each of these critical steps. So ultimately, you end up with millions and millions of motor neurons in the dish. And I call this MND in the dish because now we've got a disease in the dish which is tailored back to that individual patient. And remember, I was talking about that clinical variability problem. So just to summarise this section, so the IPSC core facility that we're establishing at the Florey Institute will provide a very powerful system to model that clinical and genetic complexity of MND. It will also provide a very relevant model of sporadic MND. So sporadic MND is the non-inherited form of MND, and this is really the gap, the knowledge gap in MND. We can't effectively model it. And we urgently need to model sporadic MND in the case of Neil and Ian, and because we need models to guide drug development. This also gives us a very powerful capacity to generate millions of nerve cells, but also other cell types that are critical for motor neuron disease. And lastly, our mission, our commitment with this facility, established at the Florey in partnership with QFMND, is to bank and provide a repository of stem cells aiming for all 400 Victorians currently suffering with motor neuron disease, so they can be part of the research program. So stage 2, so now I come back to HTS. So what is HTS? It's high throughput screening of drugs. And I'll just mention this was largely an industry-led program, but it's increasingly becoming more common in academic laboratories. And this involves the use of automation, unbiased technology, but large-scale screening platforms. And what we do in this case is we obtain drug libraries which contain thousands and millions of drugs, and we look for a biological effect. And the biological effect of interest here is promoting motor neuron survival in the dish. So as I mentioned, these drug libraries contain thousands, if not millions, of clinically approved drugs, and that's the key word, clinically approved. That means that these drugs are used, they're already prescribed for other diseases and conditions. Now this is an example of the success of HTS. So HTS, using this technology, there have been breakthroughs that have led to clinically approved drugs for cancer, HIV, diabetes, and hypertension, high blood pressure, but not for MND. And that's where the breakthrough comes. So I wanted to introduce you to this. This is the Cell Discoverer. This was launched at the Society for Neuroscience conference last year, which is the premier neuroscience conference in the world. And as you can see, this is a fully automated unit. This is the microscope here. So this is a very powerful microscope which can image motor neurons live over time, and it's matched by automation. So this is a robot which can retrieve the motor neurons from this incubator and bring them to the machine. So it's quite stunning. It's fully automated, it has a robot, and the power of this is it's able to track individual motor neurons over days and weeks and track their fate. And what we will do with this machine is we will extract multiple readouts of MND. And what I mean by this is we will track motor neuron health, connectivity, connections with other neurons, and survival in response to these drug treatments. Now that's actually me. This is me playing with the machine a few months ago, so we were fortunate to have access to a demo model. And these are the types of images one can produce. So these are motor neurons in the dish, and they're quite stunning. So you can see a lot of detail and structure, and also all of these connections between these cells. The capability of this machine is incredible. So I just have a brief video here. It's able to generate 3D reconstructions, as shown here, of motor neurons. So we get very high resolution, high detail information. And this is in contrast to older HTS systems used predominantly in cancer research, where this gives us lovely resolution. I'll just return to my talk. Okay, so as I mentioned, there is a robot. This guy actually doesn't come with it. This is what Zeiss had used in the promotion, but it's fully robotic and can automatically load plates and motor neurons And this is another example. This is a plastic plate which has 384 little wells, and each of those wells contain millions of cells and motor neurons, and you can see the magnification here. This is an example of the software, and I think this slide really sells the system. So what I've done here is a comparison between the current systems that people like myself use in the lab compared to this. So the current systems, the problems are that they are low throughput, slow going, they're restricted to what we call endpoint analysis. You have to kill the cell, they're manual, they are not suitable to large-scale drug screens. And if you have a look here, we typically use plastic plates like this, which can handle about 6 drugs a week. So that's our upper capacity. This machine, on the other hand, is high throughput. It allows you to image cells live, it's automated, unbiased, it is highly suited to large-scale drug screens, and we've estimated that we can screen about 1,000 drugs or compounds a week. And if you do the math, you get a figure like this. So we're talking about 160 times improvement in the efficiency of drug screening power, which is enormous. So that brings me to stages 3 and 4, which we should be familiar with. So these involve animal trials and human trials. We may be able to even circumvent the animal trial and go straight to humans. And this is because these drug libraries contain compounds which are clinically approved. They're prescription medications. They're already in the public domain. And what it means is we can accelerate progress to phase 2 trials. So to summarise my talk, I've introduced you to this system. This is a world-first drug screening platform for motor neuron disease, which will be established right here in Melbourne. It will allow us to rapidly screen thousands of existing drugs and compounds, which will increase the efficiency of drug discovery for motor neuron disease by about 160-fold. And the important implication of this system is that it will fast-track drug screening for motor neuron disease, and this will provide an essential massive shortcut for MND patients who urgently need effective treatments. And I just wanted to close by expressing my gratitude to Cure for MND. Thank you for having the vision and the commitment to pursue this at the state government level. And we're absolutely thrilled to work in partnership with you and operate the machine here. So thank you very much to Cure for MND and State Government and Vulcan Company.